Welcome back to today's lesson. Today we are dealing with biology, our biology first lesson, and specifically we are starting from enzymes. And under enzymes, enzymes is part of nutrition. So under nutrition, so we are on enzymes, and this is our lesson 1. So we are going to start from enzymes as we are going on and on. So now, enzymes are biological catalysts. Biological catalysts. They are protein in nature, and their function is to speed up chemical reactions in the body. So for them, and I think you know catalyst in chemistry. You check on the lessons of chemistry. You have to know to find what a catalyst is. So in these enzymes, they are biological catalysts. So they help to speed up the rate of biological reactions in the body. So now, what are their properties? How do they look like? Such things. So now, the first one, we are saying that they are protein in nature. So in nature, they are proteins. Also, they are specific in nature. It means that each enzyme can act on its own type of food. Find this enzyme is acting on carbohydrates. This one is acting on proteins. This one is acting on any other thing, but they are specific. Each one of them acts on a specific function. Then they are always from the same end. They always form the same end product. So the end products they are the same. If it is starch, the end product must be the same, and they are affected by temperature. So temperature affects them. Also, they are affected by pH, and they are just needed in small quantities. And they speed up the rate of chemical reactions in the body. Some of the chemical reactions that take place in the body, they include digestion. Digesting food is a chemical process. Also, they remain unchanged after the reaction. So after the reaction, they don't change in size, neither do they change their shape. So that is the properties of enzymes. So now we need to know how enzymes work. How do they work? How do they come to do that task? So food substrates, they are configuration structures made complementary to fit in the active sites of enzymes that work upon on them, like a rock and a key. So what does this one mean? And it is known as a rock and key mechanism. So it means they are fixed. You find it is a food substrate must enter into the enzyme, and then it closes it inside exactly, just like when you're opening a door, you insert a key, it is clearly inserted. So that is the rock and key mechanism. So you find when they are automatically fit in them. So you find the food substrate automatically must fit inside. Okay. So now the product of enzymes and food substrates are known as enzyme food substrate complex. Now the food substance is held by weak hydrogen bonds, such that at the end of the reaction, enzymes release the product and it becomes ready to react with other food substrates. So for example, on this diagram, what we are seeing that there is an enzyme that is ready to react with food substrate, and that is the food substrate. So an enzyme is like this. So now the food substrate, now it is going to enter exactly. It is going to enter exactly. So that is what we call the rock and the key mechanism. If you check on the diagram down below, the second step when it is working, you will see that now it has fixed. Now the enzyme food substrate complex, they have joined now. So now the enzyme is going to work on the food substrate. So from there, after working on it to speed up the reaction, the enzyme is now going to be set free. Its position will not be changed. It will remain the way it is, so it does not change. Then it will be now ready to react with another food substrate. So that's how the enzymes work. A summary. So you can write these notes. They are very clear how enzymes work. You can start from there. You can pause and then you resume the video as you are writing. So now let's see some of the factors that affect the rate of enzyme reaction. What are some of the things that affect these enzymes? The first one is temperature. So now temperature, we shall see how it affects pH, potential of hydrogen, surface area, concentration of enzyme, and food substrate concentration. So now we go on and we see how each of these ones affects this enzyme reaction. The first one is temperature. So now temperature, at very low temperatures enzymes are inactivated. So at low temperatures like 10 degrees centigrade, 5, their enzymes are inactivated. It means that they don't work. The enzyme activity, it increases with increase in temperature. So the reaction go fast. So the reaction goes fast if according to the temperature increase. If the temperature increases high, then that's how even the enzymes will work more and more speedy. Each enzyme has its specific temperature when it is most active. So each one has its own. Also, at that temperature, it is known as optimum temperature. The temperature when it is acting highly. However, when temperature go higher, usually between 40 to 50 degrees centigrade, the enzymes are denatured. So when the temperatures go very high, when they become very high, then between 40 to 50, the enzymes are denatured, they stop working. So they work better between 35 to 40. That's where they work better. So now here we have a graph showing the effect of temperature on an enzyme reaction. The rate of enzyme reaction, it increases with increase in temperature, but when temperatures become very high, now it again lowers. So point B is the one that we call optimum temperature. At A, the enzyme is denatured, and at C, on that diagram, at C, the enzyme is denatured. I mean, at A, it is inactive. At C, it is denatured. So now, that down you have temperature. So now this is the description which I have just summarized. You can copy if you want, but I have already summarized it. So now we have pH. So now pH, the different enzymes, they work best at different pH levels. The pH at which enzyme is most active is called optimum pH. Where the enzyme is most active, it is the one which you call the optimum pH. If the pH is very low or very high, enzymes will be denatured. If it is very low, they will be denatured, meaning they will not work. If it is very high, enzymes will also be denatured. And what is the cause of that? It is because pH changes the shape of the enzyme. Okay, pH changes the shape of the enzyme. So the shape of the enzyme is changed by the pH. You find it is changing, hence denaturing it. So now let's go on. Pepsin enzyme. These are some of the enzymes. Pepsin enzyme, it works best in acidic conditions. For example, if you check on this diagram below, we have pepsin. The optimum pH of pepsin enzyme, it is at 2, at the pH of 2, and the pH of 2 is acidic. So it works best in acidic medium. So you can see the enzyme reactivity on here, and then the pH here at 2, it is the optimum pH. As you can see, you can pause and draw. And please don't forget to subscribe to the YouTube channel to encourage me, so that we can make more and more videos. So subscribe, share the video, like, and if you have any question, or you have not understood anywhere, or you have any other thing to tell us, you can just write in the comment section just below there. So now, the amylase enzyme. Amylase enzyme, it works best at 8. So it is the active of, it is the opposite of pepsin. So at 8, which is alkaline, it's when it works better. It's optimum temperature. So now, as we can go on, these ones I've already summarized them. You can pause the video and you copy. Where you don't understand, you can ask. Now we look at another property which affects enzyme action, which is called the enzyme concentration. So now, the higher the concentration of the enzymes, the faster the rate of reaction. If the enzymes are very many and they are working on a small food substrate, then the higher the concentration of enzymes, the faster the rate of reaction, and vice versa. So if the enzymes are very many, then they will work very fast. If they are very few, they work very slow. Then we also have food substrate concentration. So the higher the concentration of the food substrate than the enzyme concentration, the lower the reaction, and vice versa. So for example, if you eat a lot of food, a lot of food, now the food substrate becomes higher than the enzyme concentration. That's when you will find that your food is not being digested. You find your stomach is like football. So please, you always make sure that you balance the two things, food substrate concentration and enzyme concentration. So that is an example which I've just given. So you people, when you are eating, you eat well. Surface area. Also surface area affects enzyme action. How? The larger the surface area for the reaction, the more quickly the reaction will go on. The smaller the surface area, the lower the reaction. That is very easy. That is easily understood. So if the surface area for the reaction is high, then the more quickly the reaction. So surface area is how the enzyme is reaching on the food substrate. The food substrate can be fixed. This one can take a lot of time. So when something is fixed, we say that that thing has a large surface area. And the larger the surface area, I mean it has a small surface area, and hence the lower the reaction, because the enzyme will not be able to reach everywhere immediately at that time. But when something is very small, it just rotates on it very fast, and then the reaction continues. So that is the surface area. So this is the lesson 4, I mean lesson 1 of our biology nutrition. So please do not forget to subscribe, share with your friends, comment, and also like the video. Also in lesson 2, we are going to begin from feeding in animals. Now we are going to look at digestion processes in the stomach. What happens in the stomach during digestion? And these are common questions in UNEB. So the five factors which affect the enzyme action, we have temperature. Second one is pH. The third one is surface area. The fourth one is concentration of enzyme, and the last one is food substrate concentration. And you should be able to explain them, because some of these things they come in practicals. So try to explain and try to understand. You know, pH, higher pH leads to lower, it denatures the enzymes. Lower pH inactivates the enzymes. That was temperature. Then pH, I've also seen it down there, the surface area, and all of these ones. So please subscribe, share, comment, and like the video. Have a nice day.